Hello and welcome back to Polymeric Biomaterials. We are still looking at structure-property relationships, and in today's lecture we are going to focus on polymer architecture and organization. So what we have done so far is slowly build up in this course, right from looking at how monomers can be joined to form short chains, and then those chains can grow to form long chains, and we have seen how placement of side groups can impact properties. We saw how N groups can impact certain types of functionality. In this lecture, we are going to focus slightly more on coarse-grained type of structures, where we zoom out a bit more and then we look at how chains can interact with each other. And we will look at different types of interactions, which we will use to classify how classify polymer architecture. Essentially, we will be looking at the linear architecture itself. Then we can look at branches that come out from the main backbone. Then you can have cross-linked polymers, or you can have 3D networks, and we will see how we can distinguish between some of them based on the extent of cross-linking, and that will lead to very different kinds of properties. We will also look at two specialized types of architectures particularly useful in biomedical engineering and in clinical applications, which are brushes and dendrimers. And then we will end with trying to understand classes of materials or polymers based on them being either thermoplastic or a thermoset or a heat set thermoset or an elastomer. So we will first begin by asking a very simple question, which is two polymers can have two polymer samples can have the overall same structure and yet exhibit very different properties, and we will look at some examples of that in this lecture itself. So you can have amorphous versus crystalline type of properties. You can have soft and flexible versus hard and rigid. So these are very different types of properties, and arguably you can have broadly the same polymer but two different samples, maybe the way they processed, maybe the way they synthesized, etc., resulting in very different properties. So this can be explained based on both intramolecular forces within and across chains, as well as chain mobility and organization, which we call architecture here. So not only are you interested now in the types of forces and interactions within chains, you are also interested in interchain interactions, what are typically called secondary interactions. And as we will see, those can also have a very large impact on specific bulk properties. And we are also now looking at segmental mobility. And to just contextualize what segmental means, you can think of a multiplicity of length scales for polymers. You can go from atoms to segments to domains to architecture to bulk. And I would encourage you to think about the order of magnitude of the length scales associated with each of these levels. There is a hierarchy. So a segment would be a small portion of the polymer itself. Domains, for example, become important in proteins, where you have some molecule that can come and bind to a certain domain in a protein. And architecture, as you will see today, is a higher order of organization, where across chains you can have covalent cross links as well, which can prevent chains from slipping past each other, resulting in certain unique properties, which ultimately result or impact bulk properties. So how all of this happens is something we will look at in today's lecture. So firstly, we will start with understanding the linear architecture itself. So the linear polymer itself is a structure where the mers are covalently linked in a long straight chain. And if you remember the noodle soup analogy or the spaghetti analogy, what you will recall is, you can think of it as a bunch of entangled cooked spaghetti. What's ironical though is the word linear itself is a bit of a misnomer, because you have to remember that there is a tetrahedral geometry around each carbon atom, right? For a hydrocarbon chain, you have a tetrahedral geometry. So the word linear itself is a bit of a misnomer, and typically when we depict polymers also, we depict them as this. To us, this would be what we call a linear, and the word linear here must be understood in the context of the fact that there is no branching in it. That is what we typically mean when we say linear. And the characteristics typically associated with linear polymers would be chain length and chain conformations, which we have seen in an earlier lecture. Now, when you think of the bonding within the chains and between chains, you can have different types of bonds. You can have what are called primary bonds, which would be the covalent bonds. And if you remember, in configuration we said polymer configuration cannot be changed without breaking primary bonds. Now, the energy associated with such bonds should tell you why that is true, right? So, these would be the covalent bonds that are used to connect the mers along the backbone. You can also have secondary interactions, which would be typically non-covalent interactions between chains, and these could include interchain interactions such as hydrogen bonding or van der Waals bonds. And you notice that the energies associated with them are much lower than covalent bond, and van der Waals is even lower. But this is a bit deceptive, because for very long chain polymers, especially linear polymers, some of these secondary interactions can become very appreciable, because you now have long chains and extensive secondary forces across the chains, and that can lead to appreciable effects on properties such as melting temperature and elastic modulus. Now would be a good time to maybe think about proteins as an example. Proteins have what we would call a linear arrangement, which we would call the primary structure. And then the primary structure itself is a bunch of amino acids connected in a sequence using covalent bonds, peptide bonds or amide bonds, and those are all covalent bonds. And then the protein can self-organize later into higher order structures. You can have a secondary structure and a tertiary structure. And what characterizes these structures, these higher order structures, are not necessarily covalent bonds, but largely bonds like hydrogen bonds, van der Waals bonds, and perhaps even ionic interactions, because proteins are charged molecules. And it's these things that determine the conformation of a protein. While the primary structure, the linear sequence of amino acids, is determined by covalent bonds, these bonds determine the secondary and tertiary structure and the folding of the protein in three-dimensional space, leading to protein conformations. And temperature, for example, can affect protein conformations, where heating can easily break these bonds, and under certain conditions they can reform and you can regain the protein conformation. That's why conformation here, that's why we use the word conformation here, as opposed to configuration, which cannot be broken easily, which is fixed once the polymer is synthesized. Again, now think about something like ultra high molecular weight polyethylene, and you can ask, you have very long chains. How do the long chains and the interchain interactions in this polymer contribute to its exceptionally good mechanical properties like high impact strength and wear strength? And it turns out that it's a combination of factors. You have long chains, then you have extensive van der Waals forces, for example, extensive chain entanglements, and crystallinity. All of these are a consequence of having these very large chains, which is justifies the epithet ultra high molecular weight. So, you see how chain interactions, interchain interactions, things like entanglements, how chains can pack together, etc., can lead to very, very good or exceptionally good mechanical properties for this polymer. Now, we look at some more examples here. I am showing you 3 different examples. Of course, one is polyethylene. We have simply listed the chain which represents the covalent bonds. Of course, you can have, as we saw, van der Waals bonds between chains. And interestingly, it's these van der Waals bonds across chains that also help give this ultra high molecular weight polyethylene its smoothness or low friction coefficients or comparatively low friction coefficients, where under shear the chains can relatively slide past each other because these are not very strong bonds. But that also means it is not very heat resistant, because even at moderate or reasonably low temperatures, the chains can start to come apart. So, you see it confers a very unique set of properties on the polymer. You also have hydrogen bonds in the polymer like nylon 6, which we saw is used quite a bit in sutures. Now, these extensive hydrogen bonds can contribute to exceptionally good mechanical properties, which make it suitable for use as sutures. You can also have dipole-dipole interactions, which are a form of van der Waals bonds. Shown here is an example of a segmented polyurethane, where we are looking at dipole-dipole interactions shown in red here. You can also have induced dipole interactions with these kinds of rings. So, you see there is a whole host of things that are going on here, where the backbone is made up of covalent bonds. You can have one type of dipole interactions here, and you can have a different type of dipole interactions here, and all of that can contribute to different properties like stability, temperature resistance, solvent resistance or solubility of the polymer, and all these things. We will now look at the next type of architecture, which is branching. So, here what you have are, you can have side chains. I have mentioned short, they could also be long. One thing to notice, these are not side groups or pendant groups that we saw earlier, they are actual chains that stick out at regular intervals, if you will. And these chains are covalently attached to the backbone, and this is again a direct consequence of the synthesis route that you take to process and make the polymer. And the way to characterize this would be to look at the number and the length of these side chains or grafts. Now, these branches, as you can immediately intuitively appreciate, will interfere with efficient packing, leading to lower density. And just as an example, you've heard of likely low density and high density polyethylene, where low density polyethylene, you will have these long branches that come out, long chain branches. You can also have short chain branches, but typically the branches are long. And in high density polyethylene, you will have very minimal branching, and even if they are very short branches. And as a direct consequence of that, you see direct differences in properties. And simply the word itself, the word density itself indicates that because of the way the chain is structured, one having long branches and the other not having branches or very minimal branches, that leads to appreciable differences in bulk density. So, lower density polyethylene will have a lower density actually compared to high density polyethylene. In terms of mechanical properties also, this one is more flexible. This is rigid, and this is less crystalline as compared to high density polyethylene, and also has a lower melting point or lower melting temperature. All of these factors or all of these properties can be ultimately linked to the presence of these branches. So, those are branched polymers. We can also have cross-linked polymers, which is the other type of architecture. We have already looked at linear, we have looked at branch, we are looking at cross-linked now. So, these are polymers where chains are connected with each other through covalent cross-links. So, they can also be secondary interactions, but the connections between chains are largely covalent, and it resembles that of the ladder structure. And the characteristics you can think of are things like cross-link density and the length between cross-links. Now, you can have multiple versions of cross-linked polymers. For example, in the dry state, you can think of rubbers, which is a good example of cross-linked polymers. You can also have hydrogels. We saw the example of sodium polyacrylate earlier used in baby diapers, which is a cross-linked polymer that swells, right. The cross-links prevent the polymer from dissolving, but also help the polymer swell. What's interesting about these polymers is the degree of cross-linking determines what type of mechanical properties the polymer can show. So, lightly cross-linked or low degree of cross-linking will typically be associated with what you would call a rubber-like or an elastomer-like behavior, whereas a high degree of cross-linking will lead to a 3D network of connections. You get a very rigid kind of a structure, and later on we will be able to correlate these to thermosets, which are highly cross-linked 3D structures, which are very rigid. Now, one way to distinguish between some of these polymer architectures, namely cross-linked versus branched, is to ask how do these polymers interact with solvents. So, if you look at linear polymers and branched polymers as well, largely they are soluble in many solvents, whereas lightly cross-linked polymers are not soluble per se, but they can swell in specific solvents. For example, hydrogels will swell in water, and heavily cross-linked polymers will neither swell nor will be soluble. So, these exhibit solvent resistance. So, that is one way maybe to think about differences between what a branching might be and what a cross-linking might be, and has to do with how solvents can dissolve one type of architecture, whereas we will find it very difficult to interact even with another type of architecture. Another example we are going to look at, or two examples we are going to look at consecutively, are examples that are particularly important in clinical and biomedical applications, namely brushes and dendrimers. Polymeric brushes are essentially chains of polymers that are attached along a 2D surface or particles at very high density. So, if they are on surface, they are you can largely think of them as a 2D surface onto which these chains have been attached or grafted. If there are particles, you can have a particle functionalized with these chains at very high density. Now, typically when these are in the presence of a solvent, water for example, which is the most common solvent encountered in clinical applications, these chains at initially low density will typically exhibit a random coil conformation, but at the higher densities you get a stretched or an extended conformation because the chains don't want to overlap with each other. Because of steric constraints, you will largely get structures that, and this is an idealized cartoon of it, but nevertheless, you will get a structure that looks like that. Now, what's interesting about these brushes is that tethering can be done depending on the surface, on the particle chemistry, can be done through covalent grafting or other types of grafting like ionic, and that depends on the type of surface that you have or type of particle that you have. And grafting can be used to change a whole host of properties of the base material. You can change adhesion properties, you can change lubrication properties, you can even create antifouling surfaces, as we saw earlier with polyethylene glycol, which if you can graft them as brushes, you can now create surfaces that now resist protein adsorption. That is what we mean by antifouling surface. Interestingly, you can also have brushes that show phase transitions, and one example of that is if you are able to take this polymer, poly N-isopropyl acrylamide, in short polynipam or p-nipam. This is a polymer that exhibits a very specific property called LCST, lower critical solution temperature. If you are hearing this word for the first time, I would suggest you look it up and try to understand what it means for you to get a better sense of what I am going to describe with polynipam. So, polynipam exhibits an LCST of around 32 degrees Celsius, and what that LCST means is it's a sort of an inflection point in some sense, above and below which the same polymer can exhibit very different properties. So, below this, let us say at 25 degrees, the same polymer can take in water, but above this temperature, let us say around body temperature, let us say 37 degrees or 40 degrees, the same polymer will expel the water and become more hydrophobic. So, that transition is defined by or is governed by this LCST. So, you can have such a material or such a polymer grafted onto a surface, and you can now control its behavior based on the temperature. So, all of these things are possible in the form of using polymers as brushes. Next, we will look at another special type of architecture that is of special significance in biomedical applications, namely dendrimers. Dendrimers can be broadly thought of as nano-sized molecules with a group of atoms surrounded by symmetric branches, namely dendrons. In fact, the word itself comes from the Greek root meaning branched tree-like structure. That is what a dendrimer is. As the name indicates, these are highly branched and largely monodispersed. So, we talked about molar mass earlier, we talked about dispersity and how different chains can have different lengths. Now, typically you get polydispersed kind of behavior when you have linear, branched, cross-linked kind of networks. When you are working with dendrimers, you can largely get fairly monodispersed products. The morphology is also ordered and it is globular largely. So, what is the dendrimer itself? Shown here is a cartoon of that, and what you see here is a structure that has some very specific subunits. So, we will look at one by one very quickly. What you have at the center is a core which forms the core of the dendrimer, and then you have these branches that stick out. And just looking at the branches, you see that there appear to be what are parent branches and daughter branches and so on. At each, when you go from the central point outward, you see that the branches keep increasing. You also see that there is what is called an N group we looked at the significance of N groups in an earlier lecture, and we saw that N group functionality is very important for biomedical applications. So you have N groups here, and just as an example, we are also showing that in the void space here you can have drug molecules entrapped. So this is an example of a dendrimer which is loaded with a drug, and what this might also remind you of is a fractal structure. If you are familiar with fractal like structures, this might remind you of a fractal structure. And you notice that all the bonds converge to a focal point, which is the core. This architecture is very unique to dendrimers, and why they are of special significance in biomedical applications is because one, the properties of these dendrimers are largely governed by the N group functionality. So as an example, you can have functionalities that are polar at the very ends in the outer edge, thus having hydrophilic groups, for example, can make this entire thing now water soluble. You can also have the N groups engineered such that they can now pass through cell membranes and deliver whatever cargo you want to deliver within the cells. And like we already said, you can have drug loading either within the white spaces as shown here, or you can also attach drugs to these N groups, and that can also be used in drug controlled release applications. One unique aspect of dendrimers is the idea of polyvalency or multifunctionality, and that is a very important idea for dendrimers, because as an example, it allows interactions with multiple biological receptor sites. But more importantly, from a practical perspective, you can also have as an example quaternary ammonium functionalized dendrimers being used as antibacterial agents. All of that comes from the ability to functionalize the N group and the property of polyvalency. If you are hearing of the word dendrimers for the first time, it is a very exciting domain, and may I suggest that you look up at this moment the use of dendrimers for drug delivery. You will find quite some very fascinating applications of dendrimers being used and synthesized in different ways and being used in various drug delivery applications. Now, we will also take a quick look at the process through which this dendrimer is prepared, because ultimately we are interested in structure property relationships, and one question you might have is, why are these largely monodispersed? And the answer to that lies in how dendrimers are synthesized themselves. So the addition of the monomers occurs in steps in a very controlled manner. So this precise control in how the monomers get added plays a very important role in the controllable architecture of the dendrimer. So you have multiple steps there, where you have the central core onto which the core is functionalized and it is activated, and you are now coupling or attaching what we call the first set of branches, which results in what we call a G1 or a generation 1 dendrimer. The G simply refers to generation 1. In fact, if you see on the earlier slide, we have shown that this is a G4 dendrimer. So that results in the first set of branches being added on in a very controlled manner to the core, and then you have specific types of activation. So this could include controlled protection and deprotection steps. These are standard approaches in chemistry, where functional groups are protected and deprotected to enable further reactions. So upon very controlled activation through some kind of deprotection, for example, you can now add the second set of branches through this coupling reaction again, resulting in a generation 2 dendrimer, and so on and so forth. You do the same set of steps again in an iterative manner, you get now a G3 dendrimer, where you have 3 levels of branching and G4 and so on. But as you can immediately see, one is this control becomes harder and harder as you go to higher and higher generations. So typically you will have some kind of a limit, practical limit to how much you can go. Also you don't want it to be very big, because if it has to get into certain spaces, it should not undergo steric hindrance and so on. So you also may not want it very big. Also practically it may be difficult to control once it goes beyond a certain number of generations. In every successive generation, you roughly get double the molar mass and the number of chain ends. And that's the key to why you get very, very controlled and very largely monodispersed structures or architectures with this kind of an approach. And typically as the molar mass increases, these molecules undergo a transition from largely an extended form to begin with, and they start taking more of a globular shape. And you will hear, as far as dendrimer synthesis is concerned, you will hear of multiple approaches such as a divergent and a convergent approach. It has to do with what is your starting point and how the build-up happens. Is it a top-down approach or a bottom-up approach kind of a thing? So again, what I would encourage you to do is, this is a very detailed topic in itself, and because our goal here is to understand structure property relationships in the context of polymer architecture, what I would suggest is you look up the applications of dendrimers in controlled release applications. You will find lots of very interesting literature, very exciting literature on this. Now, having looked at the different types of architectures, linear, branched, cross-linked, and then two special types such as brushes and dendrimers, we will now try to think of architecture from an application or a processing perspective. And we will see that you can relate these terms that we are going to look at to the terms we have seen so far. So the first one we will look at is what we will call a thermoplastic. We are going to look at 3 different types: thermoplastic, thermoset and elastomer, and you can try and see how we will relate these to whatever we have seen so far, because they all have to do with polymer architecture. The first thing to note about the word thermoplastic itself is you see the word plastic, and in common parlance polymers are often referred to as plastics very broadly, and typically when people say plastics, they usually refer to thermoplastics. Although polymers is the more general word, plastics are often used as a synonym in everyday language, everyday use for polymers, but what people really mean when they refer to plastics are actually thermoplastics. And what's important to note is that the name denotes something about the bulk behavior. So when you come across a new term like this, as I've said earlier, stop and think for a minute about what that word means, because why did they name this thermoplastic and not something else? And just that one question will lead you to a lot of very interesting insights about the word itself and what it means in that context. So most thermoplastics are mostly linear or slightly branched polymers, and the interactions you're talking about are largely 1D covalent bonds along a chain and secondary interactions such as van der Waals bonds and hydrogen bonds, etc., across chains. In thermoplastics, you typically don't have crosslinks, which means movement is very easily aided, and depending on the arrangement of chains, you can either get semicrystalline or amorphous. So you can either have polymers that can do this with a little bit of, so this would what would be called a semicrystalline material, where there is an ordered region and there is an amorphous region, or you can have polymers that are like this, where there is no order or long range order. That's what crystallinity means. So as a result of not having crosslinks, them being linear and largely governed, dictated by secondary interactions, as we saw with linear polymers, they can easily melt when heated and reshaped and reverted when cooled. They can also undergo deformation under stress, a phenomenon called creep, which we will look at in more detail when we discuss mechanical behavior later. There are also polymers that can be very easily dissolved in many common solvents. Now you can now relate how some of these polymers that we have seen so far, and think about whether they would fall under this class. I am just giving you two examples of thermoplastics. One is PTFE, polytetrafluoroethylene, which is a semicrystalline thermoplastic, and PMMA, which is an amorphous thermoplastic, both of which are used in clinical applications, as we have seen already. But then I would encourage you to think about the other polymers we have looked at and then try to see if you can classify them as a thermoplastic or not. Another categorization that we can come up with is a heat set thermoset. Again, note that the name denotes something about the bulk behavior. Now, unlike thermoplastics, thermosets are very highly cross-linked polymers, where the degree of cross-linking, as we saw earlier for cross-linked polymers, degree of cross-linking here is extremely high. And the cross-linking, because they are covalent networks, of course you will have secondary interactions between chains, but the primary rigidity that thermosets get comes from the covalent cross-links, the high degree of cross-linking. So they become rigid and the shape is permanent once cured. So curing is a word that we are using here for the process of the formation of the polymerization itself, going from the monomer, which is a runny liquid if you will, to where the cross-linking happens and the polymer is cured. And once the cross-links are complete, you get a very highly cross-linked 3D network, a dense 3D network leading to a rigid material. And the extensive nature of cross-linking or degree of cross-linking restricts the movement of chains, making them rigid. And very much unlike thermoplastics, because of the presence of these extensive cross-links, they are highly resistant to heat or melting, creep and solvents. They cannot be easily be dissolved by solvents. They are not subject to creep as much as thermoplastics, for example, and they are very resistant to heating as well. Thermosets don't have extensive biomedical applications. They are used in very specialized applications. They are limited but nevertheless used. One example of that is this idea of using them as composite resin fillings in dentistry, where you have a resin with filler particles, and typically the resin is a dimethacrylate and the filler is typically silica or some kind of ceramic, zirconia for example, for improving the wear resistance, because these are used in dental applications. And again, remember what we talked about in an earlier lecture about the N group having methacrylate. So that should give you a clue about the nature of cross-linking and relating that to the process of cross-linking and curing. So that should give you a good sense of the use of thermosets in biomedical applications. The last one we are going to look at is an elastomer. What distinguishes this from the previous two is these are also cross-linked, but the extent of cross-linking is much lower. So they are lightly cross-linked 3D networks which are rubber-like. So the degree of cross-linking is enough to permit a fair degree of flexibility. These are what would be called materials that have localized mobility and overall structural stability or rigidity. So you can have localized movement, but overall the structure will not give way because the crosslinks prevent slippage when you pull apart the polymer. And they are characterized by crosslink density and molecular weight between crosslinks. Now this is a concept we will look at later when we deal with hydrogels, where we will look at cross-linking in the context of hydrogels and how that impacts swelling of the hydrogels. We will later see that the entropic contributions allow the chains to retract or cause the chains to retract back when you pull on them. because of the differences in number of conformations, which we will look at later. But the point I am making here is you can characterize these materials both by cross-link density, and it's not very meaningful to think of a molecular weight for these polymers because it's literally one long chain. So, one way to think about molecular weight is to ask what is the molecular weight between cross-links or MC, which is again a parameter used to define or characterize these networks that we will make. So, these long flexible molecules are coiled in the natural state and stretch when pulled, but do not break because of the cross-links. They prevent slipping. Again, they are typically above the glass transition temperature, but because of the cross-links, they are unable to flow like some of the other polymers. They definitely exhibit excellent elastic properties, low stiffness and low strength, and typically are used as in applications that demand a lot of elasticity. For example, silicone elastomers and segmented polyurethanes. Silicone elastomers, PDMS is one example we saw earlier of polymers used in aesthetic reconstruction, and you can now imagine why elastomers are used for that application. So, one thing to do before we close this lecture is for you to think about how higher level architecture can be related to chain level structure, and in turn how that can be related to macroscopic properties. This is a recurring theme that we have seen in the last couple of lectures and more so in this lecture, and dwell upon that as we close this lecture. Thank you.